The protocol is used for saturation mutagenesis of a protein binding site. If a protein binding site is not known, it can be identified using this protocol. The technique has relevance for disease diagnosis and therapy. I believe its full potential in biomedical sciences remains to be fully exploited. This method can be broadly applied to any protein or reaction where desired molecules can be separated from undesired molecules. Make sure that the library is properly randomized, and make sure that during each binding and separation step, fold enrichment of desired molecules is high. It involves several steps and many details that are better demonstrated rather than explained in words. After watching this video, you should be comfortable with how to prepare a random library, synthesize a starting RNA pool, and perform a binding reaction to separate desired and undesired molecules to obtain high affinity and specific binders.
First, synthesize the forward primer and the reverse primer by chemical synthesis on a DNA synthesizer. Also synthesize a random library oligonucleotide template with 31 randomized positions. In a PCR tube, mix 1 micromolar DNA random library template, 1 micromolar forward primary containing T7 RNA polymerase promoter and reverse primer, 20 millimolar tris at pH 8, 1.5 millimolar magnesium chloride, 50 millimolar potassium chloride, 0.1 micrograms per milliliter acetylated bovine serum albumin, 2 units of Taq polymerase, and 200 micromolar each of DNTPs.